My name is Ian DeStefano. I am a graduate of the WesternU College of Veterinary Medicine in 2014. I came to Tufts in 2017 when I matched for an emergency and critical care residency here. I completed that in 2020, and then I joined the school as a faculty member. So I work primarily in the small animal teaching hospital in the emergency room and intensive care unit. In order, I think, to understand the context of sort of what we set out to do and why this study is impactful, I have to give a little bit of background. We have noted that there's a sort of rise in prevalence of multi-drug resistant organisms that cause infections. In fact, antimicrobial resistance as a whole is really on pace to be the number one health concern and the number one cause of mortality in the next 30 to 50 years worldwide. So greater than any other sort of disease syndrome that's out there, including cancer. One of the more recent, meaning in the last 20 years or so, types of antimicrobial resistant organisms that have been recognized to be out there, sort of the prototypical bacterial species that fits in that family would be something like E. coli, which a lot of people have heard of, but there are plenty of others. And what makes this subset of this group of bacteria particularly challenging is they've developed certain genes, so genetic material, that make it so that they're resistant to a wide variety of antibiotic drugs or antimicrobials, as we call them more broadly. And so carbapenems are a group of antibiotics that we use to treat certain types of bacterial infections as historically sort of a last resort type of drugs. This is not something that, your doctor would start for you if you went into the hospital with a mild problem or without really good reason. So this group of bacteria, this CRE, are actually bacteria that have obtained genes or genetic material that produce little chemicals, what we call carbapenemases, that break down these carbapenem drugs. What that leads to is in the person or animal, the patient with an infection, high level resistance to not just carbapenems, but a whole class of antibiotics that historically has been used with success to treat these types of infections. CRE has been recognized to become a problem in people, especially in the United States, in the last 15 to 20 years. And we can find these bacteria existing in the patients that we treat, and in particular cats and dogs, so companion animal species that are in our homes. All of us have some level of E. coli, for example, in our gastrointestinal system. So we know that throughout the GI tract, we've got a whole slew of different types of bacteria. Those bacteria are largely very helpful. They help with food digestion, they help with certain nutrients that need to be absorbed from our diets, but they can also be a source of opportunistic infection. And so they don't necessarily cause disease. And so what can happen, or what we know happens a lot in people as well as now companion animals, is one can become colonized by this drug-resistant type of bacteria, so this CRE, and not have any signs of symptoms of anything in particular, because it's basically just occupying the same environmental niche, if you will, in the GI tract that the drug susceptible version of, say, E. coli, as the type of bacteria we might be talking about, occupies in our GI tract. If we need to have a person or a cat or dog, a patient who we know is colonized by one of these types of bacteria, they might not become sick, but say they have a condition that may lead them to have a new infection. For example, a sporadic urinary tract infection, for example. One of the most common bacterial organisms or species of bacteria that we know causes urinary tract infections in, say, female dogs happens to be E. coli. A lot of that times, that E. coli that might cause urinary tract infection is similar to the E. coli that's found in that own dog's GI tract. And so now, if they have a urinary tract infection caused by E. coli that's CRE E. coli, now what that means is it's going to potentially take different types of drugs, to effectively antibiotic drugs, to treat that infection, and or sort of the first line drugs that we might reach to. We say, oh, the dog is having signs and symptoms of a urinary tract infection. We've diagnosed a urinary tract infection. We'll start this antibiotic. That antibiotic might not work. Part of the committee work that I do with infection control and antimicrobial stewardship here, we regularly review some of the tests that are ordered to diagnose infections, in particular, a certain type of test called a bacterial culture or culture and susceptibility test. And what this test does is takes a sample from a site that you think might be harboring an infection, say a urinary tract infection. We might take urine and send it to a lab and ask for a bacterial culture. And then the lab's job is to identify what species of bacteria is there if it grows, and then test it against a smattering of different antibiotic drugs. And that helps us as the veterinarian or the clinician, the medical team treating the patient, decide or select a drug that's going to be efficacious, that's going to help treat or fix that infection. We had started to notice a number of those culture results in sort of a dashboard that we have available to us from the laboratory that we use, come back with different types of infections that were really drug resistant to a lot of different types of antibiotics, including this class of carbapenem drugs. And so that this made us start to wonder or get worried, are these infections that we're seeing, one week, one a week later, one a few weeks after that, does this represent CRE? We worked with another lab at the University of Pennsylvania Veterinary School, and actually were able to isolate some of these bacteria and prove that they were producing those carbapenemases to kind of confirm that, yeah, what we were dealing with was CRE. We actually wrote up a paper just documenting what we did about that sort of cluster or outbreak of infections caused by this CRE E. coli. As part of our investigation, we did a whole lot of surveillance, if you will, for this type of bacteria, this drug-resistant E. coli in our hospital environment, as well as from patients that had, clinical infections that we had documented. Simultaneously, a part of that was working with the Massachusetts Department of Public Health, some folks with expertise in epidemiology and infection control, and we successfully kind of got to the root of the issue and were able to handle that and kind of eradicated that CRE from our hospital, or at least years later, I'm still unable to find that bacteria if I look around. But at the same time, there was actually a cluster of human infections. And so what happens in the state of Massachusetts is anytime a sample, whether it's from the urine or the blood or somewhere else in a patient, goes to a diagnostic lab for a culture test, if that culture test suggests that the result is CRE, that petri dish essentially has to get sent to the Massachusetts state. They basically encode the entire DNA genetic makeup of that bacteria. And the reason for that is it can help with investigations, epidemiologically to the source of these types of bacteria, because we don't think that these bacteria are commonly found in healthy dogs, cats, and people in the community, except for a very, very small proportion of people. The Massachusetts Department of Public Health uploads that genetic material into a central database that says, basically, here's the genetic makeup of this organism, this bacteria, this E. coli, where it was found, geographically, roughly, when it was found, what species it was in, was it a horse, a cat, a person? The power of that database is that when we actually uploaded some of the genetic material of the E. coli that we had looked at in our environment and had found in some of the patients we were treating, we actually discovered an epidemiologic link between the environmental samples, the animal samples, and the human samples. What the paper we wrote up most recently kind of characterizes was that there were three sort of samples from human patients in Worcester County, which is sort of the geographic county where Tufts is. It's in central Massachusetts. The patients, were unrelated. They didn't know one another. They were, treated at healthcare facilities, but not necessarily the same healthcare facilities. And so it's strange for a bug like this to just pop up in isolation, but it's more strange to have a couple of different very like basically genetically identical versions of this E. coli in a number of completely unrelated individuals. So the Massachusetts Department of Public Health had interviewed these three folks and really found no link between them, even though they had basically the same E. coli, and everyone sort of scratched their heads. It wasn't until we uploaded our genetic data and realized that the E. coli that we had isolated from our hospital and from these animals matched to these three people that also lived in the same county as our veterinary teaching hospital. So we kind of regrouped between, us at the Tufts Veterinary School, some of the folks at the Penn Vet Veterinary School, which had helped us with some of the microbiology testing, and the Massachusetts Department of Public Health. The Department of Public Health actually interviewed these folks again and asked them about their animal contact history. And as it turns out, each one of them had had a pet that had been treated in our veterinary teaching hospital during our previous outbreak. What we wrote up was kind of this timeline chronologically in this report that was published, and kind of showed that with this technology, this whole genome sequencing that allows us to, look at every bit of the DNA makeup of this bacteria, that we actually matched isolates from our environment and patients that were treated at our hospital with these human isolates. And the timeline suggests that probably what had happened was there was contamination in our hospital environment. These folks' pets had come to our hospital and had a hospitalization stay in our hospital during this outbreak while it was being managed and mitigated, and probably became colonized with this E. coli and subsequently probably passed it off to their owners that way. Meaning the only epidemiologic link between these three human cases of this identical E. coli was contact with the veterinary facility itself. While we know that there are not a ton of studies, but a few studies out there showing that animals and humans can both be colonized with very similar types of CRE, whether that causes disease or not. This type of epidemiologic link to not only human cases, but human cases where they got mildly sick from this bug was new and novel. In order to understand and help mitigate, reduce the spread of these types of bacteria in the human community, we probably need to also think about from a quote unquote One Health lens, including animal health and the environment itself as part of that investigation. Had it not been for our ability to test some of the genetic makeup of these samples in our hospital facility and the animals we were treating, and upload them to the database, there would have never been this connection to these three human cases in this rough geographic community. And so it highlights that, we really need to work together to understand the dynamics of these types of organisms and how they spread throughout the community and what that means for human and animal health collectively. We really don't know how often this happens. We, definitely have reports out there from other veterinary teaching hospitals, and I also know anecdotally of other hospitals in the US, veterinary hospitals specifically, that have encountered a very similar problem. So I wouldn't say that we have any evidence to suggest it's super common, but frankly, we really don't know. So the attention to this type of organisms, these CREs, is relatively new in veterinary medicine. In fact, we've started to survey veterinarians across the US, and most of them haven't heard of CRE before. And if you don't know that the threat is there, or when to look for it, or whether it's worth looking for it, you're not going to be able to appreciate that. What really needs to be done, in my opinion, and others that are working in this sphere, is really to invest resources, probably from sort of larger organizations, if not government organizations, which is really to determine how often it is that cats or dogs carry around or are colonized by these carbapenem-resistant organisms in the first place across the at least continental US, and whether that's different geographically, because I would hypothesize that it might be different in one area of the US versus another. But frankly, we really don't know, other than I can tell you that it's certainly not zero. I don't think it's, that this is the transfer of this bacteria is happening between people, the environment, and the patients that we treat in every hospital every day. That's for sure. But there are tons of unknowns about this type of organism. And part of the hope with publishing this report is not that this is going to save the planet necessarily, but it's proof of concept that human public health agencies really need to care about the organisms in the veterinary space in order to fully understand and most importantly, how to mitigate the spread, because there's compelling evidence that it could potentially have impacts on human health.